Hello friends, today we are going to study about protein function and we're going to study the function of a protein by binding to a ligand. So first of all, the protein function depends on the way it interacts with other molecules. Now these other molecules are known as ligands and these ligands can be another protein or any other molecules. So such protein-protein interaction is very important for a biological system. Now the ligand binds to a protein at a site which is known as binding site. So if this is a protein, it will bind to a ligand. Now this interaction is very specific. For this binding, the ligand and the site, that is the ligand binding site, should be complementary. Complementary in size, shape, charge, and its hydrophilic or hydrophobic character. Now, a single protein can have multiple ligand binding sites in which it can bind with different ligands. Now the proteins are flexible. Flexible means this structure, that is the part where the ligand will bind. This complementarity is not exactly fixed. So proteins are flexible. We already studied about breathing. What do we mean by breathing? That is the conformational change in the amino acids found in a protein, which allows for binding of a ligand, and hence a model, that is induced fit model, comes into play. That means the complementarity of this ligand binding site can have flexibility, and because of this flexibility it can change its conformation and become fit for the ligand. So it is not like that the ligand binding site should be complementarity in shape and size always. It can remain in other form, but once the ligand will come and bind to this site, the protein will change its conformation and become complementarity with the shape of the ligand so that it can bind tightly with the ligand. This model is known as induced fit model, that is the structural adaptation between protein and ligand. Now enzyme is a special case of protein function. In enzyme, the protein is known as enzyme and this ligand is called substrate. And the site, that is the ligand binding site, is known as active site or catalytic site. So it is a special case of protein in which the protein is called enzyme, the ligand is called substrate, and the site where they bind, that site is known as active or the catalytic site. Now oxygen can be bound to a heme prosthetic group. Oxygen is poorly soluble in aqueous solution. Therefore, it cannot be carried to tissues by dissolving in blood serum. And also, it cannot diffuse into tissue. Therefore, we need to bind the oxygen with some molecule. But none of the amino acid can bind reversibly to oxygen. So, for binding with oxygen, what we need? Transition element. And that element is iron. Now free iron, if it is in free form, it promotes the formation of reactive oxygen species and this can damage the macromolecules present in our body. So therefore, we need iron in a bound form. Therefore, in a biological system, this iron is incorporated into a protein-bound prosthetic group called heme. Heme is a protein-bound prosthetic group to which iron is incorporated. Let's see its structure. This is the structure of heme. So heme is a complex organic ring which is known as protoporphyrin. This individual ring, this one, this one, this one and this one is known as porphyrin ring. So iron, as you can see, present in the center which has 6 coordinates. 4 of its coordinate is bound with nitrogen of porphyrin ring and 2 of its coordinate, this one and this one, are perpendicular to the plane. Now one of this coordinate is bind with side chain of histidine and the other coordinate is bind with oxygen molecule. The binding of oxygen to the iron or to the heme converts its color from dark purple to dark red. And that's why the blood in the arteries are red in color because these are oxygenated blood, and the blood in the veins are purple colored because these are deoxygenated blood. Now these nitrogen of the porphyrin rings which are associated with the iron, it helps to prevent the conversion of Fe2 plus to Fe3 plus. Now this heme group is present inside the pocket deep which is surrounded by hydrophobic amino acid. And why is it so? Because we studied in the last video that it prevents the conversion of Fe2 plus to Fe3 plus. In Fe2 plus form only it can bind with oxygen. Fe3 plus form cannot bind with oxygen. And as because the external environment is of oxidative nature, so to prevent the conversion of Fe2 plus to Fe3 plus, this heme is present inside a pocket very well surrounded by hydrophobic amino acids. Now this heme or the iron specifically, if I say, it can not only bind with oxygen but can also bind with other molecules like carbon monoxide, NO and H2S. So the presence of heme deep inside the pocket also prevents the binding of these molecules to iron. Now one condition is called metmyoglobin or methemoglobin. What this condition is? The condition in which the iron converts from Fe2 plus to Fe3 plus state and remain in this state only and hence cannot transport oxygen. This reaction we can see in the dried blood and old meat. As soon as the blood comes in contact with the air, now air contains oxygen, so the atmosphere is oxidative in nature. So what will happen? This oxygen will bind to the iron and it gets converted into Fe3 plus and remain permanently in the Fe3 plus form only. Therefore, the dried blood and the old meat are of brown color because it cannot revert back to Fe2 plus state and for binding of fresh oxygen. Now, what would have happened if heme is not present in the pocket? In that case, if we take the example of carbon monoxide, then carbon monoxide will bind more efficiently compared to oxygen and which will lead to carbon monoxide poisoning. When heme is free, in that condition, carbon monoxide can bind with the heme molecule 20,000 times better than oxygen. But this efficiency of binding of carbon monoxide to heme get reduced because heme is present inside the pocket in hemoglobin or myoglobin, because of which the carbon monoxide cannot bind with this efficiency. Because as the heme is present inside the pocket, carbon monoxide cannot bind because of steric hindrance. Now come to myoglobin. Myoglobin, it is a simple protein made up of single polypeptide of 153 amino acid residues and one heme group. It binds with oxygen and transport the oxygen in muscle tissues. These are found more in diving animals. The reason we already studied in the last videos. Now, in the last video, if you remember, this structure of myoglobin we studied. That myoglobin constitute of 8 alpha helices connected by bends. Now, this structure only is the lower structure where each alpha helices has been denoted alphabetically. Now, in this helices, the important helices that you have to remember is one, his 93, or we can say his F8. That means the 8th position in the F alpha helix. That alpha helix, the histidine found in the 8th position of F alpha helix is bound with the heme or is bound with the iron. And another point to remember is histidine E7, which is bonded with oxygen. The 7th residue of E alpha helix that bound with the oxygen. So these two only you have to remember. Rest all we already studied in the last videos. Now coming to hemoglobin. Hemoglobin also we studied. Hemoglobin is a tetramer. That means 2 alpha and 2 beta subunit. Each subunit is like a single monomer. The only difference is it does not have D helix. Now one more thing here is if you see the complete structure of the heme, then we can also find that on the side of this heme, 2 amino acids are found, valine and phenylalanine. These are hydrophobic amino acids that helps to hold the heme in position. But these hydrophobic amino acid, they swing which is known as protein breathing, or we can say they breathe, and this breathing allows the oxygen to enter and to exit. Apart from hemoglobin and myoglobin, two other globin molecules are found: neuroglobin and cytoglobin. Neuroglobin, as the name suggests, it found in nerve cells and helps to protect the nerve from the conditions such as stroke that can happen due to oxygen radical formation. Similarly, cytoglobin, that means these are found in the cells and function similarly as neuroglobin. Neuroglobin helps to protect the nerve cells. Cytoglobin helps to protect the cells in general. Now, one question from my end for you all, that is why nature has selected hemoglobin to be a tetramer and not monomer like myoglobin. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.